Okay, so as I just mentioned, the next quiz, so you have a quiz tomorrow, which I know some of you probably have questions for that quiz, but and then the quiz after that will be chapters 16 and 17. So 16 is the second chapter of the immune system about hypersensitivity or an hyper and hyposensitivity, overactivity and underactivity of the immune system. And chapter 17 is all about diagnosing infections, which is a nice general introduction to then actually going through all of the systems and the bacteria and viruses and fungus that infect humans. We're going to focus on humans and the diseases they cause. And then when you read in those chapters, beginning with chapter 18, when you read how the infection was diagnosed, you'll have some reference from chapter 7. So today I'd like to go over 17 simply because I have that note outline. I just posted it and I just downloaded the PowerPoints from 17. So it's kind of fresh in my mind. So I just kind of want to do that one first. If those of you that are only watching the videos and not coming to the Zooms, if you would rather, when I post them online after I do 16, I'll put 16 before 17 if that upsets you because it's out of order. And then other housekeeping news. Hopefully, it looks like some of you may have started the labs. I'm not sure, but I saw some views on the YouTube videos. So I was assuming that some of you have given it a try. I have lots to say about lab at the end. But what I would say is my particular cultures are growing rather slowly because I'm growing them up in my second floor and it's not very warm up here for whatever reason. So I had intended to do day 6 yesterday on Sunday. I intended to do that and set up the biochemical test, and my bacteria really weren't grown up enough. So I'm going to try to set them up today, and then we'll be able to read the results either on what would be day 8 or day 9. So when I told you guys it would take about 2 weeks to do all the labs, the reason I told you that is because depending on how long your particular cultures are going to take to grow, you may just, if you have a super warm spot and they're growing really well, you may just plow through everything in 7 or 8 days. But if your cultures are growing kind of slowly, like mine are, I set everything up and then you have to wait usually about 2 days to get enough bacteria to record the results properly. So I'm still working on the lab report for exercises 2 through 6, because I am adding some questions from our original lab reports that we would have done if we had done these exercises in person. The gram stain lab, I'm very disappointed by what we can see with our little microscopes, but it is what it is. I think you will have had more fun using the microscope in the introduction segment and looking at the mold and the brine shrimp and the wool and the frog blood, because those structures are a lot larger than the bacteria. We really need oil immersion and a 100x objective lens to be able to see the shapes of the bacteria. So, but you can tell the colors. So that's good. That's the best we can do for now. The simple stains turned out a little bit better. And once I take photographs of them, I'll certainly share them with you guys. Oh, excuse me. But today, I would like to go over chapter 17, diagnosing infections. And I was going to just kind of follow along the handout and share screens with you with the PowerPoints. I probably will amend these PowerPoints for the way that I want them. However, I had to update my Windows, my Microsoft over the weekend. And so now the PowerPoint won't open unless I go to the website and upload a new version of PowerPoint and put it on my hard drive. So we're just going to look at the PowerPoints that are pre-made from the textbook. And again, hopefully I'm going to change them in the coming days, depending on how the lab goes. So whenever we go to diagnose an infection, the most important thing is collection. And then you'll notice that I also put transport and storage, because if it's not transported or stored correctly during transport, meaning if it has to be kept refrigerated or kept warm, then the lab test will not work. So these are just some common sites where cultures are taken or specimens are collected. Saliva, sputum, that can be with a swab, or You can spit in the tube, obviously. Blood can be drawn, urine, either clean catch or from a catheter. You can take vaginal samples, fecal samples, cerebrospinal, skin swab, right? Skin scalpel, a skin scratch, which they do with a scalpel. So all of those are places where if someone has a particular infection, where it may be harvested from, so to speak. This little tube, I thought, this little culturette, I thought was interesting because you've probably seen these at your doctor's office if they take a sample of something and they put them in a little chamber of basically nutrient agar broth or TSA broth so that it can grow a little bit before it gets to the lab. I always like to show this picture because it is the overall microbiology page. So these are the tests that would be done in the microbiology section of the clinical laboratory. In a hospital laboratory that's large enough, they would have separate sections for usually what they call, they usually have a blood typing section, and then they have a bacteriology or microbiology section, and then they have the chemistry section, which is more about running the machines for all of the chemical tests, the enzyme tests that you can get from in the hospital. But excuse me, since we're in microbiology, this would probably be the most relevant for us. All right, so I was looking for this picture. See if I didn't put it in, of course. So there's a table in your book. Here it is. It's similar to this, where it talks about the main 3 types of testing, and we divide them up into phenotypic testing, immunological testing, and then genotypic testing. And I'm going to run through these fairly quickly because of I want to keep the video to a minimum, but we could talk on and on about different layers of these different types of testing. Chemical properties. So all of the tests that we would have done in the laboratory, tests that we would ask what kind of sugar does the bacteria produce, all of those things, what kind of sugar, sorry, not do they produce, what kind of sugar do the bacteria digest, and what acids do they produce as a byproduct, all of those testings. Do they eat protein? Can they digest proteins? Can they digest lipids? I wrote down there selective and differential media, because if we look at the mannitol salt plates, or if we look at the EMB plates or the McConkey plates back from chapter 3, those would give us some examples of how you can use selective and differential media to isolate different bacteria. Rapid testing, I put the API 20 there. I don't know if they show a picture of that anymore. Oh, I guess they do. So this is an API 20, and I'm going to include this, right? So when we have an unknown in the laboratory, the first thing that always happens is it's going to be gram stained. And then based on its gram reaction, we're going to start other tests, right? And one of the classic tests to do first is the catalase test, which is something you're going to be doing in exercise 6 in your biochemical test. And that will tell us things about whether the organism is an aerobe or a facultative anaerobe. And then we can change how we're incubating it, as well as what other kinds of tests we may want to do. On a gram-negative bacteria, they are, if they're aerobic, of course, they're going to be catalase positive. And the oxidase test is one of the first tests, diagnostic tests, that helps us separate the bacteria into 2 then smaller groups of gram-negatives. Now, this is an awesome test. This is called the API 20E for enterics. And this is a set of biochemical tests in tiny, tiny, tiny little test tubes. They don't give you a size picture here, so I'm going to stop sharing for a second and just show you that that whole strip would be about this long and about as wide as my fingers. So it's made up of like these tiny, tiny, tiny little plastic test tubes, and inside of them is dehydrated media, all different kinds of selective media. So you take your bacteria, excuse me, can you turn off your microphone, Sadie? Thanks. So you take your bacteria and you incubate them in a broth culture, and then you put drops of the bacteria into all of these tubes, and you incubate it in a little water tray. You can actually incubate it only for about, you don't even have to do a whole 24 hours because the tubes are so small. Then you can read the results the next day. And each one of these sets of initials stands for a different biochemical test. No, you don't have to know what all of them are. That's not the point. The point is that when you perform this test, you're actually testing for 20 different biochemical tests in this single little test strip. This one, H2S, is one you're going to do. And I don't think any of the rest of them are particularly. And then you record scores based on whether or not it's positive or negative for each test. And there's a booklet that comes with it that tells you what color is positive and what color is negative. And then you add up the scores in these little sets. You see how the test tubes are divided into little groups of 3? So for example, in this first set of 3, only the LDC was positive, so we would have a score of 4. In the second set, only ODC is positive, so we have a score of 1. But if ODC and H2S, that would change the score to 5. You go all the way along here. These are all negative. Yellow is positive for these guys, so they're all negative. That's easy. So they're recorded as zeros. And then you literally look this up in the database, and it tells you exactly what the name of the bacteria is. It's awesome. But this particular set, these are tests for enteric bacteria. They're all gram negatives. So you couldn't use it like on a staphylococcus or a micrococcus, but they make kits for testing different staphylococcus, different streptococcus. They make different system tests like this. They're expensive though, so hospital labs would use them because it's really quick rather than making up media, full-size media for all of these tubes. All right. Now let's jump to some immunological testing. So it'll be the second block here. And the immunological testing is really advantageous for the clinical laboratory for a couple of reasons. Okay, so first you would draw blood from a person and you would separate out the serum from the red blood cells. And in the serum, that's where your immunoglobulins are. And so sometimes urine, cerebrospinal fluid, whole tissues, and saliva. And the other main advantage to an immunological method, the two big advantages are that antigen-antibody binding is so specific, and it is also super sensitive, meaning that you can't, you very rarely get false negatives or false positives, okay? So some methods, let's just look at this first. I'm trying to see if they show us an agglutination method a little bit. So an agglutination test, you can use immunological testing for an agglutination test, which is literally to see if the antigens and the antibodies interact. And if they do, they precipitate, but they don't show us, I guess they show us in this picture. And in here, right? So you could look at this under a microscope or put it into a machine and it would show how much antigen-antibody agglutination there had been by making these little particles, like a blood test, right? Where they look to see if you're A, B, or O. You can also dilute the immunoglobulin or the antigen, and you can actually do a serology test. You can do some titer testing. Now, the other main advantage of serological testing, and partly what makes it so powerful, is not only the specificity and the sensitivity, but you as the scientist, you can use the test sometimes in two different ways. You can ask the question, does the patient have immunoglobulin toward the bacteria? Or you can ask, what bacteria is this? Depending on what tools you have available or what tools you can make in your laboratory. And what I mean by that is, if you're asking, does the patient make antibodies or immunoglobulin toward the microbe, I would buy or I would prepare known antigens, proteins, or large polysaccharides from a bacteria, and I would put them in solution, and then I just have to mix that solution with my patient's serum. And if the patient has the right immunoglobulins, just these purple ones here, right, they're going to bind to this known antigen, but I would have to know this is salmonella, right? Or I suspect it's salmonella. If I wanted to test multiple antigens or multiple bacteria, then of course I would have to prepare or purchase multiple known antigen solutions. But it's super fast. You can literally do an agglutination test and you can actually see this clumping on a slide, on a microscope slide. You can also ask the question, what bacteria is this? And so this is going sort of at the opposite direction. You can see here they've streaked for isolation. We have two different colonies and they're picking one of these colonies and they're saying, okay, I have immunoglobulin to Staph aureus. That would probably be a purchase from an immunoglobulin store, online catalog. And again, you would probably have several different antibodies or immunoglobulins that would be testing. And if the person has that immunoglobulin, if the bacteria matches the immunoglobulin, then again, you would get clumping, right? Where would these colonies come from? They would have come from the patient, right? So that's also super powerful because you can say, I have serum, but I can't seem to get bacteria to grow out of the patient. So I'm going to do this test. Or, oh, it was easy. This person had like 6 bacteria in this wound. By gram staining and doing some simple biochemical tests, I know I've narrowed it down to 3 bacteria. I'm just going to get immunoglobulin to those 3 bacteria and then test, right, from my patients. Colonies that I grew. Now, you can also use super small amounts because the sensitivity and the stringent binding between antigen and immunoglobulin. And you can also label, we call them direct or indirect tests. You can have you can buy immunoglobulins that are already labeled with a fluorescent dye or a tag of some sort, sometimes radioactivity or an enzyme. Or you can actually buy, you can do an indirect test, which we'll talk about when we get to the ELISA. But we've discussed the method of the agglutination precipitation a little bit. That's just showing you these little dots here. The immunochromatography, I thought this was kind of interesting. This is showing an example using a test for Neisseria gonorrhea, I think, or Neisseria meningitidis. Gonorrhea. So it's called lateral flow tests, right? So it's like a rapid pregnancy test. You put the sample in and in fluid and it slides across a piece of paper basically, right? Where it will encounter the antibodies and then it results in a color change, whether you're positive or negative. I also listed a Western blot on there. I'm going to show this would be an indirect Western blot because on the blot itself, we would have separated the proteins, a target protein, and we're asking, does the patient have immunoglobulin to that protein? And in this case, it does. We call it the primary antibody. And then, but the antibodies are clear and we can't see them on a piece of paper. So we apply a secondary antibody that is specific for the FC portion of the primary. And to that secondary antibody, we attach a label. In this case, it's a color label. You can see it's kind of light purple to dark purple. There are orange labels as well. You could also label with a fluorescent marker. You could also label with radioactivity. The radioactivity is difficult, becomes a little bit more difficult because it's a problem to dispose of it. And the fluorescent dyes also usually have some specific disposal concerns, so you can't just pour them down the sink. So a lab may choose to use this colorimetric, which is an enzyme substrate, and I think this is horseradish peroxidase. So it's a commonly occurring enzyme in nature, so you can just throw these in the trash or wash the excess antibody down the sink. This is using immunofluorescence, and in this case, it's a direct test. So what they've done is they've purchased immunoglobulins to syphilis that have already been labeled with fluorescent marker, and this is the patient's serum. They're mixing them together, and rather than looking for agglutination, they're going to put this under a UV light, a fluorescent microscope, and they're going to be able to see the actual bacteria lit up here. These are the individual bacteria because they've been coated with the immunoglobulin that you purchased that was labeled, right? If the person doesn't have syphilis, then those immunoglobulins, remember, antibodies are super specific, those immunoglobulins aren't going to stick to anything, and this would be a dark background, be blank. And then last but not least, the ELISA, and I would like to go through an indirect ELISA test with you, a little bit step by step, and again, we can review this if it doesn't make a lot of sense, but I put down as an example, I put down an ELISA is the second test for, well, actually it's the first test, the screening test for if you're positive or not for HIV. And so what is done is the sample, we're going to see a person who's positive here in the well A set and a person who's negative here in the well B set. And you purchase the little plastic dishes with the antigen already absorbed to the well. Now, those dishes are going to be, let's see if I can get a whiteboard here real quickly. They are going to be relatively small. And I'm just going to draw one. They're rectangular shaped like this. And they would have 12 little wells across the top here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 little wells there, and they would be numbered 1, 2, 3, 4, 5, right on the plastic, all the way across. And then they have, I believe it's 8 rows down, A, B, C, D, E, F, G, right? 1, 2, 3, 4, 5, 6, 7, H, A, B, C, D, E, F, G, H, like that, 8 rows down. So there's 96 little wells. And if you looked at this container from the side, right, it would be skinny like this. And we would see the wells, we would just see the wells depressed like this. Right, so from the top, they look like little circles, but they're actually little plastic wells, and they probably actually go a little deeper than that, more like that. So what that drawing is, they're showing you 2 wells, right? One with a person that's going to be positive for our test, and one for a person that's going to be negative for our test. And as I mentioned, the test is going to be for P24. That's the antigen that we're looking for. And in this case, this antigen, P24, this is a protein on the surface of HIV. So if you are infected with HIV, you're going to be positive for this test. If you're not infected, if you have hepatitis and you don't have HIV, then you're not going to have antibodies that bind to this first protein. So this is an indirect test because we are, oops, I lost my other share here. Because we are, this would be the P24 antigen, the known antigen that's absorbed to the well. Now, if I was a researcher and I had to do this test from scratch, I would have to find purified P24 and put it on the Petri, on this little well, these little plastic wells. And by incubating it, it would stick to the plastic well. If it's a commercially available test, like the test for P24, I would just purchase the plates and they would already come with the antigen attached to them. Then I'm going to put in the patient serum, and the patient serum in this case is going to be someone who's positive for HIV and someone who's negative. The person who's positive, their immunoglobulins are going to bind to the antigen. Remember, it's super specific and it's a super tight bond. So I can rinse this out with some buffer solutions, and it's not going to dislodge these immunoglobulin antigen bindings. But in the well over here, notice I didn't have the right immunoglobulin because remember, this person has something else. They don't have HIV. So if the first immunoglobulin doesn't stick, the primary immunoglobulin, then the second one can't bind because there's nothing to bind it to. That's these light green ones here. So we add our second antibody or our secondary antibody that is conjugated or bound to, in this case, it's going to be an enzyme. And that's this little white shape that's attached to the tails of these antibodies. Now, where would I find, where, how would I do that? Well, I would either purchase it, or you can actually, it's a pretty simple chemical reaction. You can actually do it yourself in the lab in like an hour. And then you would rinse out the excess. And in the case of the person who's negative, since they did not have the primary antibody bound, the secondary antibody doesn't bind. So there's nothing for the enzyme isn't going to stick. Then we have the substrate, and it's an enzyme that we chose because when the enzyme binds to the substrate, it changes the color. And we don't have, oh, here's a positive ELISA test. That's a little better than my picture that I drew, right? And you put this little plastic dish into a machine, and it can actually read, you run controls. This isn't a good example because the controls would be in decreasing concentration either down the plate or across the plate. So they'd be super dark yellow at the beginning, and they would get less and less concentrated. And those controls, then those standards, we would call them, they will make a standard curve. And then you can compare by a computer, basically. You put the plate, it goes into a spectrophotometer, which can read, not this time how turbid the solution is, but how much yellow is in the solution. And it's going to tabulate that and then tell you exactly how many microliters of immunoglobulin or picograms of immunoglobulin this person has in their serum. So it's a much more informative test because it's more quantitative than doing agglutination testing. So, all right, so that's the ELISA. And then there are some genotypic methods that I'd like to run through with you because they are even more specific. And so you're going to use DNA rather than, and the advantage of them is usually you do not need a pure culture first. So it's a faster test because you don't have to actually grow the bacteria up and then, or the virus, or collect a lot of serum from a person. You can just do it directly. So I listed hybridization first, and this uses small probes of either DNA or RNA that are known to be complementary to specific sequences of nucleic acid isolated from a particular microbe. Ugh, that's a mouthful, right? But basically what that means is you would have to have, get another whiteboard here. You would have to have a known sequence of DNA or RNA. Let's do DNA, A, T, C, G, G, whatever, right? Dot, 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 dot. And let's say this sequence, this particular sequence is super specific for Salmonella. It's a special sequence for Salmonella. Meaning it doesn't show up in E. coli, it doesn't show up in any other organism, right? I'd underline that. So then what you would do is you make a probe, which is, I'll make it a different color, I'll make it red. That's like a complementary messenger RNA or a complementary DNA. So let's make it DNA. So it would be, the probe would be TAGCC, right? And these probes can be quite small. They're usually a little bit longer than this. They're usually about 20 nucleotides. But then this probe will only bind to Salmonella because they are complementary. So you would purify a bunch of DNA from Salmonella, heat it up so that it's separated into two individual chains of DNA, and then you would add this probe. And if the probe matches, the probe has usually has some sort of a tag on it. Like I'll put a little star here for it would be radioactive, or it would have a fluorescent molecule on it, and you could shine UV light on the culture or the system, and it would glow. So it's kind of simple. That does take a little bit of time. This is these are some standardized tests. The FISH test, we call it fluorescent in situ hybridization. This is nice because you can actually confirm the diagnosis right on a piece of glass or sometimes a biofilm. And for example, you would put, so here's a positive blood culture tube. So you would have the bacteria already gram stained, and you want to know if they're Staph aureus or not, like if they're MRSA, for example. If you had a particular a probe of nucleic acid, either DNA or RNA, that was specific for MRSA, and it had a fluorescent tag on it, you can literally just put this on like by dropper, wait 5-10 minutes, rinse it, and then if this DNA matches that DNA, it's going to glow, and you can see it under a UV fluorescent microscope. They listed microarrays here, and I would like to go through microarrays real fast, and we'll come back to PCR. So microarrays are these little diagnostic chips. They're not super common yet because companies are still trying to decide what DNAs they're going to put on the chips or what messenger RNAs they're going to put on these chips. But they can load basically DNA or RNA that's unique from multiple organisms onto these little, they're basically plastic micro wells. And then you put your DNA down from the patient and shoot it with lasers, and these fluorescent markers light up, and then you can tell, a computer basically will tell you what person has, what disease they have. Super expensive at this point, which is why they're not done for every little piece of disease. Whole genome is low, so low. The cost of the whole genome is low enough now that people can start to use it. And it is used, noticed for rapid analysis of outbreaks and drug-resistant organisms, right? This is becoming more common, but you do have to have the capability for genome sequencing, which is still a little bit expensive. So you would have to send this out to a laboratory that had that. And pulse gel field electrophoresis is a sort of specialized DNA fingerprinting. It's very similar. There's a picture of that. This one here. So it can be a little bit faster. So you would have your organism, your unknown bacteria, and you would put it into, you'd collect it from a isolation plate, and you put it into this melted agarose tube, and then you lyse all the DNA, and it by restriction enzymes. First you lyse all the bacteria to get the DNA out, and then you treat them with restriction enzymes. But rather than looking for small pieces that are cut, you're looking for large segments that are cut. And so what they do is they load it into an agarose gel and then run the electrophoresis, and they keep alternating the field of the electrophoresis so that they use the electrical current to separate out these large blocks. And then you can look for commonalities. You would run a control, and you can look for, oh, look, patient here doesn't have that particular DNA, that this particular block, but patient 3 does. So this person has whatever organism this profile represents, Staph aureus or whatever. All right, so I was going to talk about PCR real quickly here as well. And there is a picture in your textbook. This would have been covered in chapter 10, which we did not cover chapter 10 in its entirety. So I was just going to talk about PCR a little bit here. And it's relevant as well because that's what the type of testing that's being done for coronavirus. PCR stands for polymerase chain reaction. And this is a really cool technique that was developed by a couple of guys actually in sort of out in the Pacific Northwest. I think they were in Idaho. And what they did was they took some DNA and they said, you know, if we could find a way to very quickly separate DNA and we could make unique primers that could bind to that DNA, kind of like the probes we talked about earlier, we might be able to find a way to, the main advantage of PCR is what it does is it amplifies or multiplies rare pieces of nucleotides. And what I mean by rare is not only they're unique, I should say rare, unique pieces of nucleotides or fragments of nucleotides. And so in order to do this, you have to already have some things in your toolbox, I usually say to my students, right? So you already have to have a toolkit that includes a set of primers. And these primers, we're going to start with DNA primers. And the primers are going to be complementary DNA sequence So they're known. We already know what they are. They're complementary DNA sequence to the organism that we're interested in isolating or we're trying to identify. You also have to have all the nucleotides. You would buy those from the supplier, chemical supplier, meaning you would have to have A's and T's and G's and C's. You also have to have a special enzyme. You have to have a special DNA polymerase. It's called Taq polymerase. And it is a DNA polymerase that can withstand high heat. Okay, so if you have all those things, you're ready to go to do PCR. When you're developing a PCR test, one of the key things, one of the big difficulties is here, constructing these primers, because you have to already know the sequence of whatever it is you're trying to amplify. So for example, with the coronavirus, why this test wasn't available immediately was because first somebody had to sequence the whole genome of the virus, and be sure that they got it right before the primers could be developed. Then you can either purchase these primers or you can make them yourself in a DNA synthesizing machine. And then the PCR reaction itself is this beautiful, simple reaction. So the first thing that happens is you would obtain some DNA. So this is going to be my DNA here. And I would have gotten this from my patient, from my bacteria, right? If I grew the bacteria, or from my patient. I think in the rapid test, they're just using saliva. So you're spitting into a tube. So there's going to be DNA from the virus in that tube, as well as DNA from all of your cells. We're not interested in those. So the primers are going to be specific for the virus, right? So what you do is you collect the DNA and you lyse the cells so the DNA is released. And then you put it into a container at 95 degrees Celsius, not Fahrenheit, Celsius. And at 95 degrees Celsius, the DNA will, all the hydrogen bonds between the two chains of DNA will release, will dissolve. So we're basically going to be doing replication without all the extra enzymes. So we don't need helicase and gyrase if we just heat the DNA up to 95 degrees Celsius. It's a cell-free system. Then the next step is that you're going to add the primers. And the primers need to stick at a lower temperature. We call that annealing. And the primers, why I told you they were a set, is because remember DNA has an orientation, 5' to 3'. I'm doing a bang up job there, and 5' to 3'. And remember, when replication happens, when DNA polymerase works, it needs to have that primer on there from RNA, from primase, in order to get started. And so we're skipping that step as well. We've already got primers, one set that's 5 to 3 and one that's 3 to 5 that are specific for this particular sequence. So I think I'll make those red. Well, I started with purple, so I'll make them purple. So we have one primer that will sit here, complementary 3 to 5, and then another primer that'll sit here in this direction, that complementary 5 to 3, 3 to 5, right? And their sequences are going to be different because they're going to be complementary to the DNA here. So we get the primers to stick. So we anneal. I think that's usually at about like a 60 degrees Celsius neighborhood. 60. And then the cool thing is we turn the temperature back up to about, this black, turn the temperature back up to in the 70s, like let's say 72 degrees C. And the primers will then finish, oh, I shouldn't draw it like that. The primers will then elongate with the Taq polymerase. They're going to elongate and make a complementary copy of this original piece of DNA. So I'm going to draw it, try to make it a little more accurate here. Switch to purple. All right, so this would be my first new strand, complementary and anti-parallel, and here's my other one. Right, so now I have not one copy of this really rare, hard to find piece of DNA, but now I have 2 copies. And this is the genius of PCR. Are you ready? You load the system with enough materials so that you can just repeat. And all of this, all of these steps, you can do it in about 90 seconds, minute and a half. So you can repeat it a lot of times really fast. And what that does is it gives you multiple copies of this very hard to find piece of DNA in amongst all kinds of other junk DNA. And it's super specific. It can't mess up because it's only for the organism you're looking for. When they say real-time PCR, that has an added feature that the primers are labeled, which is what some of the testing is right now, real time. That we usually identify that as qPCR because it's quantitative. Whoops, sorry, I forgot the C. And we don't call it RT because that stands for something another type of PCR. That stands for reverse transcriptase PCR. So baby Q PCR equals real time PCR. And the advantage of that is when you label the primers, now each of the new sets of DNA, right? Each of these is going to be labeled with my blue balls there, and I can read those. They're fluorescent dyes, so I can read those with a machine that can detect fluorescence and increasing amounts of fluorescence, which is cool. And the only other type of PCR that I want to briefly mention is RT, which stands actually for reverse transcriptase PCR. I think they usually put a dash. And this is utilizing one of the enzymes, so RT for reverse transcriptase. That's an enzyme from HIV, actually, that can take messenger RNA and transcribe it into DNA, double-stranded. We call it complementary DNA because in humans it will be missing all the introns, so it's just the coding region. And then this complementary DNA, keep that blue. This complementary DNA, we can then put into a PCR reaction like that, right? And so in essence, rather than looking for DNA, we can look for messenger RNA, which is particularly useful when we're looking for viruses, because many viruses, remember, have RNA as their genome. So, right, I'll type that down next. Right, so if RNA is their genome and they have to make messenger RNA as well so they can make all their proteins, this type of PCR is particularly useful for looking for viruses that have an RNA genome. All right, I say that's enough of chapter 17. If you want more details on any of those things, let me know because I can give you more, but I'm trying to keep it relatively short so the recording doesn't just like last for hours. All right, so